Now the second chairperson for the session is Dr. Surajit Ghatak. Dr. Surajit Ghatak is Professor and Head at Department of Anatomy, AIIMS, Jodhpur. Sir has graduated and post-graduated from Armed Forces Medical College, Pune. He has authored 2 books in anatomy and has numerous international and national publications to his credit. He is also a Fellow of International Medical Science Academy and a Fellow of Royal Society of Biology, London, UK. He is also a life member of Anatomical Society of India and member National Academy of Medical Sciences, New Delhi. He is also Dean of Students' Welfare and Chairman of a Students' Ethical Committee. We welcome you, sir. Sir, we are really grateful that you both have spared your time to be a chairperson with us, because we all know that it is exam session and everywhere exams are going on and everybody is busy there. Now our prestigious guest speaker for this session is Dr. D.R. Singh. Dr. D.R. Singh, everyone knows him, and he is a very renowned teacher of anatomy. Dr. D.R. Singh is Emeritus Professor at Department of Anatomy, King George's Medical University. He is an author, outstanding researcher, and a meticulous teacher. Sir did his graduation and post-graduation from King George's Medical University. He did his PhD way back in 1982 under Dr. Mehdi Hassan from AMU. He headed the Department of Anatomy, King George's Medical University from year 1990 to 2000. He also headed the Department of Anatomy at BP Koirala Institute of Health Sciences, Dharan, Nepal, and Nepalganj Medical College. Sir has authored 7 books, and one of his recent books, the second edition of that book, that is Essentials of Anatomy for Dentistry Students, have been translated into Turkish language also. Sir is Founder Fellow of Anatomical Society of India. He has been awarded with Hari Om Ashram Alembic Research Award by MCI. He was awarded Dr. Dharam Narayan Memorial and Dr. Mehdi Hassan Gold Medal. He has obtained fellowship from Indian Academy of Neurosciences, and he has been awarded a prestigious Dr. Sukhbir Prasad Jan and Dr. Shamsher Singh Memorial Oration by Anatomical Society of India. He has got more than 50 years teaching experience, and his love and passion for teaching is unparalleled. It's always a pleasure to hear you, sir. I welcome you, sir. Sir is going to deliver his talk. Sir is going to deliver his talk on ergonomics and anatomy. Good ergonomics, you all know that the good ergonomics is that which is compatible with human anatomy, as this reduces the risk of musculoskeletal disorders, and whereas bad ergonomics is responsible for several discomforts like headache, pain in shoulder, low back ache, pain in neck, etc. So now I invite Dr. D.R. Singh to present his talk. I will request Sir that he will receive a pop-up to unmute himself. So please accept that invitation and unmute yourself. And I will request you to share your screen. Over to you, sir. Thank you.
Professor Jyoti, I would request you to please allow me at the account DRDRS to share.
So you have been allowed. Are you able to share your screen?
No. So, good morning, Professor Jyoti. Dr. Singh would like to share his screen from another account. So if you can allow DRDRS account to share the screen, please.
Okay, okay. So we'll do that. We'll do that. Just a minute. So it has been done.
Thank you.
So welcome you. Thank you for being with us, sir.
Okay.
So we are able to see your screen now.
Okay.
Screen of Dr.
Just a minute.
Yes, sir.
The screen is sharing already, sir.
अब आपकी स्क्रीन शेयर हो रही है? एक बार ट्राई करिए. नहीं हो रही? So we have allowed you to share your screen. So we have allowed you. Unmute Dr. Adivasi. अभी सर unmuted नहीं है. So we are able to see you and yes, sir. Yes, sir.
Now, good morning, everyone. Chairperson, Professor A. Sharif, Dr. Punita Manik, Head of the Department of Anatomy, Professor Jyoti, and other members of the team. I would like first to thank Dr. Jyoti and her team for giving me this opportunity to present my talk today. My thanks are also to Dr. Punita for including me to be a part of this conference. As some of you might know, I spent best period of my life at King George's British College, now upgraded as university, and I completed my MBBS in 1964 from here and had the privilege to join this college right after that as demonstrator of anatomy, and then I served this period, as Dr. Jyoti has mentioned, till 2000. Having spent more than 40 years in this college, I learned the subject of anatomy from my revered teachers like late Professor Dharmnarayan, Professor A.C. Das, Dr. A. Halim, who taught me most of the part of the anatomy, and I just dedicate my talks to these teachers as well as my past and present students. Dr. Jyoti has already introduced me, and I would therefore not spend too much time on this page and would like to directly jump into today's topic. My topic of today is ergonomics and anatomy. And this is a topic that uses the applicability of anatomy as a subject. And it is very relevant to for today's lifestyle that we have been forced to adopt. It is important to understand the real meaning of ergonomics and its application with the anatomy of human body. As some of you might know, ergonomics comes from two Greek words meaning work plus laws of. Ergonomics is the study of man with respect to his work, and that's why ergonomics is also known as human engineering. It is important to understand the ergonomics application in improving our day-to-day life. The tasks we do, the equipments we use, and the overall job environment. There are no specific ergonomics regulations, although OSHA, which is Occupational Safety and Health Administration under the United States Department of Labor, continues to mention ergonomics injuries while working. It studies the relationship between the employees, his job, and the environment to that job. So I hope it is now clear what ergonomics is and why it is called human engineering. I would like to draw your attention on this slide of a multipolar neuron. The blue part that you see around the nucleus is the basophilic part that you would have studied in histology at the light microscopic level. The same is visualized as endoplasmic reticulum in the right picture, and ergonomics utilizes this information at the cellular level quite effectively. I am sure you realize the applicability of ergonomics in various disciplines as depicted in this slide, like physiology, anatomy, engineering, medicine, anthropology, psychology, and biomechanics. And I am also certain that experts from all these fields can relate to the importance of ergonomics in their respective areas. Let us now look at various types of ergonomics. And it is basically divided into three categories: physical, cognitive, and organizational. Let us first look at physical ergonomics. For someone like me, who has always studied and taught anatomy, I find this type of ergonomics the most relevant to my field. Physical ergonomics is about the human body's responses to physical and physiological work. Repetitive strain injuries from repetition, vibration, force, and posture are the most common type of issues and have various applications. Physical ergonomics also deals with the physical load on the human body when performing activities like sports, jobs at home, or dealing with any product. The contribution of basic anatomy lies in the improving fit between people and the things they use, ranging from the hand tools to the aircraft cockpit design. And physical ergonomics defines this relationship quite beautifully. Let us now look what is cognitive ergonomics. As the name suggests, the cognitive ergonomics is the field of study that focuses on how well the use of a product matches the cognitive capability of the users. As you know, the cognitive means conscious intellectual activity such as thinking, reasoning, or remembering. And I am sure all of us have used cognition during our student days. We have all given theory examinations, and then all those vivas that we gave were all based on the cognition of our subject. The most important aspects under cognitive ergonomics include mental effort, decision making, interaction with computers, human reliability, and work stress. Lastly, the third type of ergonomics is organizational ergonomics. Organizational ergonomics combines elements of both the physical and cognitive domains, and this is the newest category of ergonomics. The goal of organizational ergonomics is the attainment of a fully harmonized work system that ensures employees' job satisfaction and commitment. It includes managing everything in an organization to make it a better place to work. It also includes the study of technology's impact on human relationships, processes, and overall institution. Organizational ergonomics also looks at driving overall efficiencies at the workplace. How well the monitors are placed, whether the desks and chairs are comfortable or not, how can we maintain better health and hygiene of the employees, all this falls under the organization or office ergonomics. And therefore, the working in the office does put some health hazards that we all need to be aware of. In case the proper posture while working is not followed, we can very quickly start feeling its impact on our body. Whether it is the height of the monitor, your chair and sitting posture, how do you bend your wrists, how well you place your feet on the ground, all these are important aspects that need to be followed. As you will look in the slide, the correct and the good postures are shown while working with a computer. There are some typical conditions involving our bodies which result due to bad ergonomics, and these are collectively grouped as office syndrome. They consist of redness in the eye, the hyperemia, headache, shoulder pain, back pain, pain at the wrist, so-called carpal tunnel syndrome, and the shoulder pain. So these are some of the common conditions which may be present either isolated or in the different form in an individual who is not adopting the correct posture as advocated in the ergonomic principles. Ergonomics is the science of adopting workstations, tools, and equipment which are to be compatible with the human anatomy so that the musculoskeletal disorder injuries due to ergonomic stressors, they are reduced. In other words, we can say they fit the job to the person rather than person to the job. Here I am showing some of the incidences which are most common in the pain and how much incidences is there. For example, we can see that the maximum incidence is present in the low back pain, the lumbago. Then the pain is in the knees, 46%, pain in the wrist joint, about 43%, and shoulder pain, just also close to 42% to be more precise. Now this slide shows the ergonomic sitting posture, and here you can appreciate that the three important things which are indicated in the red: the top of the monitor, which should be at the eye level and slightly tilted. The back of your pelvis should be slightly forwards so that you are more your hamstrings than on your tailbone to relieve tension on your neck and shoulders. And also equally important is to keep wrist straight and elbows close to the side. The fourth thing which one should keep in mind is the flat fit. That means studying an individual sitting posture in the office, back and neck position at the work, and positioning of their leg. They are the more important points which everybody should remember. In this slide, I have tried to show the effect of posture when we are standing. The first figure shows a good posture. The other figures, they are depicting some deformities. For example, the hollow back, a flat pelvis, a slumping posture, a very straight so-called military posture, and a rounded shoulder. Correct posture puts least amount of strain on our muscles and joints, whereas the other type of postures, they are producing effect on our health. This slide is also showing you the poor posture and the good postures, and incorrect and the correct posture, especially when the ladies are using their standing posture. Effect of posture is also seen besides standing when we are sitting, and a bad posture, as you can see on the extreme left, and a good posture. The right figure shows that the slouching, slumping, and other type of posture, they can provide discomfort, and neck pain, back pain, and the wrist pain are the common discomforts. Problems caused by the bad sitting posture, they include these four important structures: joint pain, particularly in the hip, knees, and ankle; muscle stiffness; permanent slumping, as you see on the extreme left; compression of abdominal GIT organs; and sitting in a slouch position all day negatively affects our lung capacity and causes difficulty in breathing, which is quite important these days due to the pandemic COVID-19, which we are all afraid of. The anatomy of sitting is posture essentially depends on the relationship of body's posture and the gravity to the chair, and a far posterior position on this and a well balanced on our chairs is shown in this slide. So, because we have all seen that the correct posture is very important to maintain our health, I have shown here a typical ergonomic chair, which I am sad to state that most of the offices that I have visited do not give enough attention to focus on this. And an ordinary chair is provided to the workers, and a good chair is very important to ensure that proper ergonomic principles are followed and the correct posture is maintained. Anyhow, this is an ideal ergonomic picture, and you can see the various points highlighted here with the dotted lines. I will not go into the details of individual points. The best ergonomic chair should be provided for the proper health of the individuals. Now we should understand why what is the correct ergonomic sitting posture in the office? People are asked to sit for a quite handsome time, and sitting for a long time regularly provides many health issues. Correct sitting posture is even more important. However, many of get this wrong, and no support for the curve for the lumbar spine, excessive strain on the lumbar disc, the support for the footrest, and our hands, they don't reach the mouse and the keyboard, and we sit too far forward. So these points should be avoided to maintain the correct ergonomics. Now this slide, I will draw again your attention to the subject described under the science behind our gloves. And as you know that our extremities, lower extremities are maintained for the balance of the body weight and the stability, whereas our upper extremities, they are more maintained for our free movement. Our hands and wrists are not intended to bear our weight. And so it is no wonder that many of us experience pain or discomfort when doing exercises like yoga poses. I do not condemn that yoga has many advantages also, but we should not start putting our weight on our wrists, because it can provide discomfort, and therefore too much strain should be avoided. This slide also shows many so-called pilot positions in different yoga poses, which I am not very aware of. And just to summarize, putting full or partial body weight on our hands results in pain or discomfort. Now, if we look a little closer at the anatomy of our hands to increase, this lady is shown and to put her entire body weight on her wrists, and further aggravation is caused by doing many repetitions of wrist stretching exercises or holding body weight poses for a prolonged duration. So now this slide shows a slumped posture in the two individuals marked with the red, and a correct posture here. And as far as possible, you should avoid slumping posture and should maintain the correct posture for the health of your spine. This is the period when we are all passing from work to home situation. And as you can see in the upper series of diagrams, the extreme right, a person is lying on his bed and is performing the work from home, as most of us are doing in our offices. Whether too much work, too little work, or even the work from home, they are the wrong postures. The lower series of picture, they have the right working postures, they are shown. The same thing we should apply, not that we should sit for a very long time, and the so-called static posture is not desirable. We should maintain a shifting posture, and incorrect posture and correct postures are depicted in the lower diagrams also. As far as possible, we should try to maintain the correct postures. The same thing when we are having the wrong sitting posture, a correct sitting posture is shown. And when we are, say, for example, in our lecture classes, we are standing for at least 1 hour per day, the correct standing position should also be maintained. So, I just like to give some take-home messages from you. Slouching shoulders while seemingly relaxing. It appears from the figure that it is quite relaxing, but This leads to stress and tension in the various muscles. Bad posture also causes chest breathing and low body oxygen. Some of the diaphragm, since the diaphragm provides nearly maximum oxygenation to our arterial blood due to the gravity, blood flow at the bottom of the lungs is about 6 times stronger than at the top due to the reduced oxygen level. Indeed, people who are slouch always have chest breathing since their abdominal organs are compressed. Their chest breathing also prevents normal lymphatic drainage of the lymph nodes and from all these organs which are located near the diaphragm. This is another vital function of the diaphragm which should be maintained. So we should avoid a slouching shoulder position which appears actually relaxing but is not. It causes many health results. The same thing again depictive. I will not go into the details here. The sitting on a chair, the level of the monitor, the position of the, you should ensure that your feet are flat on the floor and knees are in line of your hips. These things should be maintained. Now, this is a slide which I have just taken, that the average human beings spend more than 55% of their working time, that is nearly 8 hours of working time, in the sedentary behavior such as sitting in our offices or wherever we are working. For people who sit most of the day, their risk of heart attack is about same as in the smokers. This is a report from the cardiologist from the Mayo Clinic. So we should understand the proper positioning or the advantages of the proper posture, and we should avoid all these factors which can cause some discomfort to our various body parts. In the end, I would like to thank all of you for listening to me, and a special thank to you, all my teachers and graduate, particularly the undergraduate students who are sitting here. I just finished my talk with this, that maintain a good posture and enjoy a good health. Thank you very much.
Thank you so much, sir. Thank you so much for pointing out the importance of good posture while working, because we all are doing very long days jobs. And I like your saying that fit the job to the person rather than person to the job. That was a catch line. So I request Dr. Sharif to take over the charge of the session. Dr. Sharif, we have sent you a message to unmute yourself, and I would like you to chair the session.
Thank you, Dr. Jyoti. Thank you, sir, Dr. D.R. Singh, for such an illuminating talk on the ergonomics, and which is highly relevant in these times where most of the people are working from home, and probably the working conditions at home may not be as optimal as those in the offices. So I think it is important for people to take the message that you have given during this talk, and then also take care of their own bodies, and as well as understand the anatomy behind the ergonomics. So ergonomics is probably more noticed when it is not there. If something is not ergonomic, then only you notice it. That means if you are doing your job and you don't notice anything wrong, that means your environment and your equipment has been ergonomically designed. So, sir, there are some questions. So, one of the questions from Dr. Ghatak is footwear also an important part of ergonomics, especially amongst armed forces? Hence, footwear designer should keep in mind the ergonomics involved rather than on the fashion aspect. Sir, would you like to add anything, sir, to this, Dr. D.R. Singh?
No, I fully agree with what has been said. Or, Dr. Ghatak, I am thankful to you for just. I have seen your good posture while I visited your institution, and ergonomics is perhaps very important during the designing of these spaces. So I fully agree what you have seen, and I have nothing much to add to this.
Sir, I would like to extend. Do you have something to add to the ladies' footwear, where the very high heels and
I
stilettos?
would just like to advise that the high heels should be avoided as far as possible, because the high heels, they put a lot of discomfort on this. But somehow this is the era of the designing and fashion, and ladies will not agree what I advocate to them, because the high heels, every lady, and especially the young girls, they are fond of purchasing the high heel sandals and other.
If they are using for extended periods of time, what could be the likely problems, sir, that they may have?
They will get a strain on their ankles, and later on their fight and especially the hamstring muscles can be just become stiff, and in later part of their life, when they put off their shoes, they might feel some discomfort when they come from their offices or from just their market or whatever places they visit, because these ladies are also the working ladies, and they once wear such type of shoe wears, they are just for a prolonged period quite sufficiently. And what I have shown in my last but one slide, that the Mayo Clinic has advocated that the most of the time, 50%, 55% of our time we are spending on our work, and so that it should be avoided as far as possible. So there are many other factors which the ladies are conscious of, and it cannot be just said that because perfectness cannot be achieved. What we have studied in the ergonomics, the ideal principle, even I am sure that once I taught my whole career in the Department of Anatomy, and with you at Dharan also, I was not adopting all these principles which I have mentioned today. So it is very easy to put up to tell some instructions to our audience, but very difficult to follow the same principles.
Sir, during histological classes and teaching and learning, do you think that some ergonomic principles have to be adopted?
It cannot be followed actually in the true sense, because most of the working tables we have to see in the microscope, and we have to bend our back just to see what the student is being shown. Maybe that at some better institution like yours, the student is provided with a comparison microscope, and instead of just going down in the microscope put on the table, he can see the particular slide on the adjoining screen. So that can adopt a good standing posture, but that is not applicable to most of the places. And what the majority, because many institutions, they are not even getting the proper positioning, the proper table, proper chairs, they are not provided. And probably I fully agree that they cannot be provided. Ideal situations, they cannot be provided by most of our institutions due to our lacking funds.
Still, we can urge the responsible heads of the departments to
and we should hope for the best.
Yes, sir. Yes, sir. Because histology is one place where extended periods of time we keep peeping into the microscope.
Yes, quite true.
Yes, sir. So, are there any more questions? Sir, I have not received any questions, sir.
Okay. Thank you, sir.
Yes.
Thank you so much, sir.
Thank you.
Thank you, Dr. Sharif. We really thank you to take out your time from your busy schedule and that you chaired the session. Thank you so much, sir.
I thank you and Dr. Punita and all the organizers for giving me this pleasant duty of chairing a session by our dear Professor Dr.
Yes.
D.R. Singh, with whom I have been associated for at least 2 decades, I think.
Okay, okay.
Yeah, from 2000 onwards.
Okay, so I didn't know that.
So very nice listening to you, sir, and I'm very happy that I could get the opportunity to chair your session. And this is one subject which I am also very passionate about. Thank you very much, sir. Thank you. Thank you.
My pleasure.
Thank you, sir.
So now, on behalf of Team IVACON, sir, we express our gratitude and appreciation by presenting you a certificate. Sir, I will send you the certificate.
Thank you. Thank you.
Thank you so much, sir.
Okay.
Thank you so much, sir. Sir, we were really blessed to have you here, and your passion for teaching is really unparalleled. And I think you are a best example of a lifelong learner also, because even after retiring, you have learned so much about technology that have popped up with the passing decades, and you updated yourself for everything. Thank you, sir, for being with us. So now we will move on to the next session. Before that, I have an announcement to make that we are going to create breakout rooms, but the participants will be able to join it after the completion of this session, that is 10:30. We will announce when they have to join the breakout rooms. They will be created, but you will be able to join it once we announce for that. Okay. So now I welcome Dr. Sabita Mishra, who has already joined us. And I would like to introduce ma'am. Dr. Savita Mishra is Director, Professor and Head at Department of Anatomy, Maulana Azad Medical College, New Delhi. She is magnificent researcher and very admirable teacher. She did her undergraduation from SCB Medical College, Cuttack, and PGDNB from UCMS, New Delhi. Then she did her PhD from AIMS, New Delhi, the institute which she later joined. She is in the department with a fetal repository. Ma'am has edited Langman's Medical Embryology. She has reviewed all the 3 volumes of Cunningham Dissector and also revised and edited Inderbeer Singh's textbook of human histology. Ma'am has organized several workshops and conferences like International Conference on Stereology in 2016 and 18, workshops. She has several administrative responsibilities too. She is Now, are you able to hear me?
I can't hear you at all. I
Uh,
can't see anything, can't hear also anything. Suddenly, I don't know. It's just, I've missed everything.
okay, I think the system, some problem occurred. And once I started introducing ma'am, I think nobody was able to hear me.
No, I couldn't hear anything.
Okay.
Neither could
So
I hear anything.
Just I will repeat the.
Should I? No, I have not. I can only hear you. I can't see anything. Should I enter again?
No, ma'am. No, ma'am. Have you shared your screen? I'll just allow you to share your screen.
Okay. I can't see actually. The Zoom thing has also gone. It's just disappeared from my screen. So that's what I thought, that probably I should log in again. I can hear you,
Okay.
but I can't.
You can't see.
I
Okay,
can't
ma'am.
see anything.
Okay, ma'am. So just log in again, ma'am. I would request you to log in again.
Right.
Okay.
Yes, yes.
And if all the participants were not able to hear me also, so for that sake, I would like to just go through the introduction of Dr. Sabita Mishra. Ma'am is Director, Professor and Head in Department of Anatomy, Maulana Azad Medical College, and she is magnificent researcher and admirable teacher. Ma'am has done her undergraduation from SCB Medical College, Cuttack, and done her PG DNB from UCMS, PhD Anatomy from AIMS New Delhi. She is teaching anatomy for almost 30 years, and her special interest is in neuroscience, developmental anatomy, histogenesis, and stereology. She has established fetal autopsy lab where they do regular fetal autopsy, and they have also established a fetal repository. And that is a very good source for research. And she has edited Langman Medical Embryology. She has reviewed all the 3 volumes of Cunningham Dissector, and she has revised and edited Inderveer Singh's textbook of human histology. Ma'am has organized several workshops and conferences like International Conference on Stereology in 2016 and 18, workshop on embalming and embalming, plastination and museum techniques, histological techniques, body donation seminars, and fetal autopsy workshops. She has got several administrative responsibilities too. She is chairperson of Bureau of Indian Statistics for Anatomy and Forensic Medicine. She is convener of Specialty Board of Anatomy for National Board of Examination. So I would request ma'am, has ma'am joined?
Yes, I've joined.
So I would request now ma'am to share your slide. Share your slide and enlighten us with the recent advances in molecular regulation of neural tube defects. फरहान, हमारी इससे फिर हमें लॉगिन कर दो, इससे हमें लॉगिन कर दो।
ये हैंग हो गया था, अच्छा बहुत हीट हो गया लैपटॉप।
अभी तो शुरू हुई बस से
नहीं, ये बहुत
Actually, I'm not getting the share button.
Ma'am, at the bottom panel, there is a green button, slide show.
That green button is not there, the video. I can't see my panel.
नीचे चले गए हम शुरू का, मुझे कहते हैं नीचे टास्क बार में देखिए जूम होगा, उस पर क्लिक करिए।
मैम नीचे टास्क बार में एक बार थोड़ा सा ट्राई करके देखें, शायद वहां से दिख जाए।
टास्क बार में पार्टिसिपेंट ज्वाइन, It's not showing, it's only showing the audio's icon.
Yes, yes,
It's showing. This is Dr. Sharif's. I can see mine. I can't see at all. Okay, start my video. Okay, now you have given me.
Now you're with.
Can you see my screen now?
No, ma'am, we are able to see you.
Okay, you're not able to see my screen?
No, not now, ma'am.
Okay, just a minute.
मेरी meeting यहाँ से करो, रुको.
Can you see me?
Yes, ma'am, we are able to see you.
So what's going wrong? I actually, in the previous one, I could see my share screen, but now it's somehow. Now I can see this thing. Just a minute. I don't know what's happening. Really sorry for all this, but the share button is not coming actually.
Ma'am, can you see more?
I can see, I can't see anything actually. I can just see a plain, the taskbar.
No problem, ma'am. We will play your video.
I'll just try to see because I can't see anything. Just a minute. Join record the computer reaction, invite participation. The share screen is not there.
Ma'am, can you give us a screenshot of your screen, whatever you are seeing? ये screen share करते हैं. यहाँ से.
I'll just. Shivani, please ask Jyoti, more screen. More screen share. I can't share my screen. Just check. This is. Okay, can you see my screen now?
Yes, ma'am, we are able to see your screen.
Okay, I'll just go to the full screen now. Oh, I'm extremely sorry for all this glitches that happened at the beginning.
No problem.
Am I audible?
Yes, ma'am, you're perfectly audible.
Okay, so at the outset, I would like to thank the organizers, Dr. Punita, Dr. Jyoti, for giving me this privilege to deliver a lecture in this international online platform. And the topic of my lecture is Contemporary Neuroembryology and its Relevance in Fetal Medicine. I feel extremely honored to speak after the Doyen of Anatomy, Professor D.R. Singh. Sir is a legendary in anatomy. I'm grateful for the chairpersons, Dr. Sharif and Dr. Ghatak. So neural tube defects are one of the most common congenital defects which are encountered in India. Though worldwide, the incidence is 0.5 to 2 per 1,000 established pregnancies, especially in India, North India, it's 7 pregnancies per 1,000 established pregnancies. When the fetuses are born, some of them are incompatible to life. They get aborted very early in gestation. Those who live, do not even cross infancy, and those who survive, it's a lifelong disability affecting the quality of life. The picture out here is one of the pictures that we had in our department, the fetal autopsy workshop, which we had. In MAMC, we do a lot of fetal autopsies. In the last 3 years, out of the 100 fetuses we got, and the last year, in fact, because of COVID times, we didn't do any collection. We have got about 34 neural defects, which would say 1/3 of the defects were neural tube defects. So what is the scope of fetal medicine in terms of neural tube defects? There are a number of challenges in this, but the precise molecular techniques and scanning have helped us in diagnosis very early in gestation, and that helps in utero interventions by in utero surgery or fetal medicine, thereby helping these infants to have a normal delivery. But of course, prevention is always better than cure. So before we go, I'd just like to give the statistics of the time of gestation versus risk of birth defects. It's known that the first 2 weeks of gestation are lethal. Any teratogenic insult at that time will not be the fetuses will not be able to survive. The period of organogenesis is between 3rd to 8th week, where major congenital anomalies set in. And later on, from 9th week to term is when, if there is any insult, it will lead to morphological and functional abnormalities. With this timeline, let me walk you through the development of the embryo from the very beginning, post-fertilization. The morula, a ball of cells, we all know, a spherical shaped cells which have got arranged into an inner cell mass and an outer cell mass. And by the time it reaches the uterine cavity, it has all got shifted to one side, and a cavity develops. We all know it's the amniotic cavity, and the cells are aligned in 2 layers, an upper layer and a lower layer. So this is the first time that the embryo is having a dorsal and ventral orientation. With the dorsal and ventral orientation of this flat disc of 2 layers, the cells which are present on one side of the embryonic disc, the epiblast, are called the anteroventral epidermal cells, which would be the ones responsible to form the cranial end of this embryonic disc. So these anteroventral epiblastic cells, they express genes, transcription factors, which are OTX1, LEM1, and HXK1, which are secreting 2 factors, lefty and cerebrus, which inhibit the nodal at the primitive node and form the cranial end of the embryo. The absence of lefty and cerebrus are going to form the caudal end of the embryo. So once this embryonic disc is formed, we have to understand that it is going to have a cranial end and a caudal end. So before that, the embryonic disc, which was a flattened disc, has formed a slipper-shaped disc, as we all know. And in the center, from the caudal aspect, the primitive streak grows to the rostral part, and the swelling out here in the rostral end is called the primitive node. In that, there's the primitive pit. At this time, the entire embryonic disc is secreting BMP4, which is a transverse TGF, it's parts of the TGF beta family. This TGF beta, this BMP4, along with fibroblast growth factor, has the capability of ventralizing the entire mesoderm. So the entire mesoderm of the embryonic disc at this time will become completely ventralized to form. It will be forming the intermediate mesoderm and the lateral mesoderm. But dorsalization of the mesoderm is required. For that, the primitive node, which is secreting caudin, noggin, and follistatin, is going to antagonize the BMP4, thereby causing dorsalization of the mesoderm. The cranial end forms, and the notochord, the dorsalization will form the notochord, the somites, and the somatomeres. So the upper part of the embryonic disc is dorsalized by these three factors, while the caudal end is dorsalized by the brachyury tube. It is important to understand, along with the neural tube, the surrounding mesoderm. That is the reason why I was giving these examples. So once the embryo is formed with the craniocaudal axis, the primitive streak has formed, which is giving us the caudal and the cranial aspect. From this cranial aspect, now we have to understand that for the body axis, the embryo has already developed its dorsoventral axis. It has developed the craniocaudal axis, and the third part which the embryo has to have is the right and left axis, right and left orientation of the embryo. So this primitive node secretes nodal, and this nodal is responsible for high concentration of serotonin on the left side. For high concentration of serotonin with FGF8 help in secretion lefty2, and this lefty2 is actually responsible for secretion of the PTX2 master gene. And this PTX2 master gene is responsible for keeping the secretions on the left side. It doesn't transfer it to the right side. The organs which are going to form the left side are actually all have the PTX2 master gene. Therefore, the heart, the stomach, they will be all having the PTX2 master gene. So median axis is being formed, and this primitive node would be giving the midline structure, mesodermal condensation, which is going to form the notochord. The midline genes which are responsible are the SSH, secreted by the notochord, the lefty one, and the ZIG3, which is a zinc transcription factor. These are all the midline genes which are maintaining the median plane of the embryo. So the embryonic disc, as the mesoderm is formed, the central part of the primitive node would be going rostrally. As we all know, it forms the notochord, which is first with the hypoblastic cells, they form the notochordal plate, and gradually as the hypoblasts get replaced, it forms a condensed part, which forms the notochord. And the low picture also, one can see this green structure that you see is the notochord. The overlying part of the ectoderm changes into neuroectoderm, and the underlying part is the endoderm. The mesoderm moves laterally from the primitive node, and the part of the mesoderm which is immediately adjacent to the notochord is the paraaxial mesoderm, which is going to give rise to the vertebral column of the developing neural tube. And cranially, it will be the cranial somatomeres which are going to form the cranial cavity. Thereby, along with neural tube, it's important to understand the mesodermal condensation which are going to form the bony cavity in which the neural tubes are going to lie. And SHH is secreted by notochord. So the slipper-shaped disc which was there, it's getting, the notochord induces the overlying ectoderm to become the neuroectoderm by induction of SSH. And what happens is that the slipper-shaped disc lengthens. And as it lengthens, this lengthening of the body axis along also the neuroplate takes place by a mechanism which is called as convergent extension. What is convergent extension? In this, what happens, cells which are going to form the neural tube, neural plate, they move from the lateral to the medial direction. So as they move from the lateral to medial direction, they lengthen in size, they widen in size, and they help in lengthening of the neural tube. This is maintained by a pathway which is called, or it's regulated by a pathway which is called the planar cell polarity pathway. So we briefly just go through the steps which involve the neural tube development. Is the neural plate is the ectodermal placoid over the notochord, which lengthens along with the body axis by convergent extension. As it lengthens, the lateral edge of these neural plate, they fold to form neural folds. The neural fold will fuse first in the cervical region, and this fusion proceeds both cranially and caudally. The cephalic and caudal end of the neural tube communicate with the amniotic cavity anteriorly by day 25 and posteriorly by day 28. Completion of the neurulation, the closed neural tube, is completed beyond that. So now let's see what are the molecular factors which are involved in formation of the neural tube. So convergent extension caused cells to move from this is the neural plate overlying the notochord. So the overlying ectoderm has got differentiated to form the neural plate. And in this neural plate, with the increase in SHH, the BMP levels are very low. So as the BMP is decreasing, is being decreased by the SHH, and this along with caudin and noggin and follistatin are forming the neural groove, and the neural groove would get opposed to form the neural tube. Just before closure would be coming out the neural crest cells, as we have all been taught. But the caudal part, the SHH is low, and the caudal part is by the neural tube is in response to the transcription factor WNT3A, and the patterning of the entire neural tube is by the homeobox genes which are maintained for patterning, and it is regulated by retinoic acid, which is higher on the cranial end and low on the caudal end. The neural crest migration takes place just before closure. So when there is low BMP, it forms the neural tube, the ectoderm. When there is intermediate BMP, the neural cells migrate out as the neural crest cells, and these neural crest cells along with FGF and WNT are responsible for the migration. On the dorsal tube of the dorsal part of the neural tube, there is another factor, PAX3, which is important in cell migration, which is called the second wave of migration, which helps the cells to migrate to the final destination. There's a dorsal migration and a ventral migration. Dorsal migrations are they pierce the basal lamina to go into the form the melanocytes, while the ventral migrations will be coming up into the pharyngeal arches, as we all know. I'm not going into the detail about this. So now let's go on to the molecular regulation of this neural tube bending. So the neural tube bending which happens is different from the upper part and the lower part. As you see, this is a picture showing a CV stain pictures on the upper part and the lower part. The picture A and picture B, the picture A is on the higher part of the cranial part of the neural tube, while picture B is the caudal part of the neural tube. And let's concentrate on the schematic diagrams. If you look at the schematic diagrams, in the upper part, you see that there is a V-shaped bending. There is only one bend out here, which is called the median hinge point. The median hinge point, the BMP2 out here, the SHH in the upper part is very high. As it is very high, it is also secreting noggin. The noggin is active in the upper part. And here the BMP2 is also acting on the noggin to inhibit it. As a result, the noggin is not impressing on the BMP2, and because of SHH, there is only a median bend, which is called the median hinge point. And this median hinge point is the one which is present in the upper part, that's in the cranial part of the neural tube. And as the neural tube would be closing, it is going to form a convex mold for the brain to grow. While the lower part, where the spinal cord will be forming, there is another bend on the lateral side, which is called the dorsolateral hinge points. Here what happens, the noggin is, the SHH is less, the noggin is also less. So the BMP2, which is activating the noggin, is again having an anti-antagonist effect on BMP2. As a result, another fold is being formed out here, which is called the dorsolateral hinge points. And this dorsolateral hinge points, as they form, they will be enclosing the spinal cord. So we can see that the spinal cord is, when we come to the low part of the neural tube, there are two hinge points. One is called the dorsolateral hinge point, and one is called the upper part has only and the median hinge point, while the upper part of the neural tube only has the median hinge point. And this has been seen across all species. Now let's look at the mechanism of this neural tube closure. As you look at this picture, this is the neural tube. We have already done the elementary part of the neural tube closure, but now we have to go on to the detail. So as the neural tubes are coming close to each other, the ectoderm, which was on the lateral side, then after the neural crest cells have migrated, they are also getting separated out. So as a result of this separation, but they will start growing over the neural tube and become separate from the neural tube ectoderm. So the neural tube is going to get submerged inside, and these cells which are outside are going to form the non-neural ectodermal cells, which are responsible for forming the epidermis. Therefore, what is the take-home message is that if BMP is not antagonized, it is all going to form as epidermis. So once the neural tube is formed, this is getting separated from the neural tube. And one can see that there are a number of mechanisms by which it happens. First, they come opposed by these markers out here, and then there is slight protrusion of the cells. These transcriptional regulators are the grainy head transcription factors. They come and they oppose with each other by forming these processes, and there are a number of transmembrane proteins which help in fusion. While the neural tubes, when they are fusing, they would be also coming closer. We'll do in the next slide. But now let's look at the cells at the median hinge point. In the median hinge point, if you see, you can see that the cells out here have changed their shape. They become pyramidal in shape. This is due to the convergent extension, where there is a apical constriction of the cells with basal round nuclei. So there is apical constriction, there is interkinetic nuclear migration, and this all happens by the non-canonical WNT PCP pathway, which is the polar cell polarity pathway. While above at the dorsolateral hinge point, it is opposite. The nuclei are basal and apical simultaneously, as you can see here. This is the median hinge point structure, and this is the dorsolateral hinge point structure. And the factors which are responsible are the PAX3, CDX2 on the dorsal aspect of the neural tube. So the molecular mechanism is that as it is happening, this as the neural plate is bending, there are a number of factors which are responsible for this median hinge point. The apical constriction is by these apical actin filaments, which are under the control of these markers, Schrum, Vinculin, and Marx. While the neural tubes is developing, the neural cells are also proliferating. So there'll be proliferation of these cells by these factors, which are HES1 and NUM, while when they are coming and closing, the formation of this closure, there's a lot of apoptosis also going, which are under the influence of caspase and BCL10. And the neural crest migration is taking place by the activity transcription factors SITE2 and twist connection. So once the bending has taken place, these folds are coming close together and the neural tube is closing. So initially the neural tube, when it is formed, we all know that it has just a revision of what we have done, which we usually study is that it has 3 layers, a marginal layer, a mantle layer, and a outer, so an ventricular layer, a mantle layer, and a marginal layer. So this is the layer where there are neurons present, and it is a pseudostratified epithelium, which later there is a lot of cell proliferation, migration of these neuroplasts to their to their destination by neurite outgrowth. They will form synaptic contacts and form their final destination. So let's come to the neurulation. Till now, whatever I did say was all about the primary neurulation, that formation of the neural tube, the formation of the neural groove, and closure of the neural tube, which we have studied, is all primary neurulation, and it ends at the second sacral vertebra. But just below that, at the sacrococcygeal junction, secondary neurulation takes place. The cells which are present out here, the part at the caudal end of the embryo, which is the primitive streak part. The primitive streak is the caudal part of the embryo, which regenerates as the notochord grows, and finally once the structures are formed. But the caudal eminence gives rise to these cells, which finally luminize. They become neural in character. They enclose the lumen and form the neural tube, with the notochord being there in the ventral region. So as this is inducing, there is a caudal eminence which forms the neural tube, and this neural tube would go and align with the neural tube above to form the neural tube incomplete. So the primary neural tube and the secondary neural tube are becoming continuous with each other, and it has been seen that both the neural tubes defects in closure would be the one responsible for neural tube defects. Anything going wrong at the secondary neurulation probably is responsible for a sacrococcygeal teratoma. So the closure of the neural tube is also very interesting. I just made a very fast talk about it. But once we see that how it closes, it doesn't close simultaneously like, but it has they have closure points. So the closure points are called closure 1, which is between the cervical and the hind point. So this is the first part where closure takes place in the schematic diagram that you see. And what happens in this, as closure 1 is taking place, this goes both cranially and caudally. The closure is like a zipper, a fastening of a zipper which goes up and down. As it goes cranially, there is another closure point which is seen in mice but not in human. This is just caudal to the anterior neuropore, and more rostrally there is another there is another closure point which is called closure number 3. So there are 3 closure points in mice: closure 1, closure 2, and closure 3. While in human there are only 2 closure points. One is called closure number 1 and the other is called closure number 3. Why is it important to understand these closures? It's only when there are defects with this closure point that one would have cranial defects. We will say neural tube defects. If the closures are not complete, one would have closure, one would have neural tube defects. So this closure points would be the ones which, when they are open, they would give rise to a total open neural tube as it goes anteriorly. If it comes onto the anterior part of the neural pore, this part is not closing, then it will give rise to a part where the front of the neural tube is not, the cranial part of the neural tube doesn't grow, giving rise to an anencephaly. While if the rostral part, this part 3 is very rare, very uncommon. If this is present, it's going to give a split anencephaly along with the split facial structure. All these experiments have been done from mouse models which stimulate the embryonic models. So an embryonic 10 and a human 35, it's almost equivalent stages, and you can see very nicely that the mice embryo shows 1, 2, and 3 de novo closure points, while the human embryo shows 1 and 3, there is no 2. But in experimental animals, they have seen that if closure 2 is not present, no neural tube defects are detected. That means probably over evolution, it has been realized that neural tube closure number 2 is not relevant. This shows a scanning picture of the neural tube closure. And if it doesn't close, the neural tubes are too wide and the neural plate is too short, what would happen? They will not be able to fuse, thereby leaving defects like this. So neural plate, when it's too wide for neurons to fuse in the midline, this is because of the failed convergent extension in a wide neural plate and a short anteroposterior axis, which is because of the PCP genes, planar cell polarity pathway involving genes. So this is a picture which we are all familiar with, showing an embryo from a round disc to a flattened disc, and then it comes to as an elongated disc. And as it becomes elongated, you can see by this picture that you see with the red margin, it's a completely open neural tube. Only at 5th somite stage that they would be starting to fuse. And you can see like a zipper of a coat, or we can say that it looks like coat buttons which go up and down. They don't go in any. So this is the way they are closing. So as they close, we have the anterior neuropore and the posterior neuropore. We all know that this closes by 25 and 28 days respectively, having the brain and the spinal cord. So once the neural tubes are formed, we have to have the neurons, and for that one needs neurons in the spinal cord. So we'll be having dorsal horns and ventral horns, and we know that the sensory neurons which are present in the dorsal horn are under the influence of high TGF and low SHH. This causes, so we are talking about the molecular regulation at the spinal cord. These are under the influence of PAX3 and PAX7 genes, and when they are present, they form sensory neurons. While on the ventral aspect, we know in the ventral aspect there is high, because of the notochord is very close to it, there are high SHH with low TGF beta, and the factors which are responsible are NKX2 and NKXC. They are the ones which influence the neuroblast out here to form ventral motor neuron cells. So similarly, if you look at the brain, we have the expanded cranial part of the neural tube is forming the brain. So we have the brain has the prosencephalon, the telencephalon, the diencephalon, and the myelencephalon, and then the mesencephalon, and we have the rhombomeres. So we have the forebrain, the midbrain, and the hindbrain. And if we look at this, we have two organizers. One is called the anterior neural organizer, or the ridge, which is present on the rostral part of the forebrain. This is called the anterior neural ridge, and between the rhombomeres and the midbrain and the hindbrain level, this pink area that one sees is called the isthmus part, which is again another organizing center for the brain. So throughout the ventral part of the brain and the spinal cord, the inducer is always SHH, which is being secreted by the, you can see the brown color out here, which is for the brain and the spinal cord. Now let's go for the expression of genes, and in the hindbrain genes, we have forebrain, midbrain, and hindbrain. So this, if we see the hindbrain, the actually, sorry, somebody is just interrupting the slides by making some figures. So I would ask him, please do not scribble on the slides. So there are two organizing centers, as we see, which are making the brain to differentiate into the forebrain and midbrain and hindbrain region, and these are the transcription factors involved. It has been seen that the LIM1 and OTX2, as we had said before, are probably only present in the human brain, which is also because of evolution, a big frontal lobe. So it is known as the new head, or the, which is much more advanced in human beings. So the ventral part is always induced by SHH and the dorsal by BMP. So if there is a failed neural kinase, this is a picture showing between mice and human. If there's failed cranial neurulation, what happens? It's open. So along with the brain, the skull flaps are open. There is first protrusion of the neural tissue, and this neural tissue, which comes and gets exposed to the extra embryonic environment, the amniotic cavity, gets degenerated, and thereby there is an anencephaly. And the similar kind of picture is also seen in human beings. So these pictures that I have shown here are all from our fetal autopsy lab. We do regular fetal autopsies for clinicians, for the obstetricians, because in therapeutic abortions where they want us to correlate their ultrasound findings with our autopsy findings. Many times we are right, many times there are differences between the ultrasound findings and the autopsy findings. So the first case that I'm showing is a failure in the cranial neural tube. We've all seen this. This is an anencephaly. So when there is the closure 1, or the cranial part of the brain, when it is exposed, because the skull vault is also not formed. So the degeneration of this neural tissue which comes out is getting degenerated, and therefore there is no brain. When it happens in the rostral brain, it's anencephaly. When it happens in the midbrain, it's merocranium, and when it's happening in the posterior brain, it's called holocranium. And the findings are usually determined by ultrasound. The latest level ultrasounds which are there, level 1, level 2, they would be able to pick it up. and usually the mothers are advised for termination. The meningoencephalocele, this is a primary defect of the cranial mesodermal defect. If the mesoderm is opened at the skull, at the occipital level or at the frontal or the parietal, then the meninges herniate out. If there is only the meninges, it's a meningocele, but when there is brain with it, it can be a meningoencephalocele. So these are failed neural tube closures. There may not be any neural tube closures, but because the skull mesodermal defects are there, it will be causing these kind of protrusions from the neural tube, spinal cord, or the brain. This is an interesting case that we received this year. This was a fetus. It showed as if there was a big meningoencephalocele. We did report it as meningoencephalocele, but when we looked at it very carefully, we could see that there are actually two cerebral cortexes. And in the literature, when I did a literature survey, we could no way find that there is an evidence that there are two cerebral hemispheres. It's not there in literature, but what has happened is also something which we were quite confused, and we tried to see the literature. They said that the rostral part of the brain, if it doesn't grow, then probably you can have cranial defects, that the baby had absolutely normal, a normal face with no problem in its appearance. So probably it was an occipital bone flap missing. So the skin flap is normal, the brain has herniated out and protruded so much because of the expanded space that it has grown. We did histology of both the parts of the brain, and both of them look like occipital cortex, so which is very interesting. So I've not put the histology pictures here, so which we have to decipher what's happened, but it is most likely an meningoencephalocele. When there is an extensively craniorachichisis totalis is when the spine neural tube does not fuse at all. So there is a closure defect at closure 1. It remains open from the midbrain to the lower spine. There is disruption in the convergent extension involving the PCP pathway. So there is a short rostrocaudal body axis completely incompatible to life. Spina bifida, as we all know, this is a neural tube defect at the lower part of the lumbar region. And in this, normal embryos, they have vertebral arches developed. We know that they develop from the lateral sclerotomes, which move dorsally to close the neural tube, and this cartilaginous part becomes bony. But if the bony part, the neural tube does not fold, remains open, then they are not able to oppose and close the neural tube, close the vertebral arches, thereby leading to spina bifida. So spina bifida is it can be a vertebral defect in secondary to the neural tube defects. So if there is a neural tube defect, it's open, the vertebral column cannot close posteriorly. So this kind of structures, when there is with the meninges, with the neural tissue, we call it a meningomyelocele. These are issues which can be treated in intrauterine life, like a myelocele, a very small lesion in the posterior part of the lumbar spine, and it has a bifid lumbar column or a vertebra throughout. And these are also can be incomplete closure of the neural tube, but if they are detected early, probably one is able to do some kind of surgical intervention, intrauterine, only in specialized. So similarly, if there's a very small spinal defect, sometimes they come with lipomatous lesions. And in these, because of the lipoma and all that, you can find that the spinal cords are tethered to the adjacent structures, and that would be causing sometimes pulling of the spinal cord and leading to hydrocephalus also. And sometimes these structures, these tumors, when they have been seen exposed, they can have a multipotential germ cell nature. Sometimes it's seen that the central canals are also dilated, called the hydromyelia. This is a very small lesion, a peduncular lesion at the posterior part of the lower lumbar region. And these can be actually treated. If they can be treated, then probably we'll be saving these babies having lifelong disabilities. And due to midline defects, sometimes we see that these are some of the pictures that these are from our own autopsy lab, where we had a single holoprosencephaly. These are midline defects. And you can see that there is no cerebral hemisphere. It's only single cerebral hemisphere with the brainstem. And the eyes are also very close with the proboscis. This is also when we've had about 3 to 4 holoprosencephaly in our labs, and these are midline defects, and it's actually a high dose of alcohol drinking in the pregnant mothers at early stages, when they do not even realize that they are pregnant, actually kills the midline cells and causes this. So there you have these kind of defects, midline defects. This is a semilobar prosencephaly, where the frontal pole is only fused while the rest of the cerebral hemisphere is separate. And you can see that the close eyes, eyes are very close together. This is again, this fetus when we received, it had a big meningomyelocele, a vertebral defect, spina bifida. Upper part had no defect, but when it was dissected out, one could see that the cerebellum had protruded into the cervical region. Therefore, it's an Al-Chari syndrome, where the cerebellum goes into the neck region, and that is because of pulling of the brainstem by this increased pressure. So there are number of transcription factors in the midline which maintain these midline. This is the HNF3 beta, and there's overexpression of goose codin causes malformations at the cranial end, which would be causing duplication of the blade of the face also. And if there is a sacrococcygeal teratoma, that's because of if it fails to regress, the primitive streak usually regresses. If it fails to regress, it gives a big sacrococcygeal teratoma, and these also can be treated surgically. So along with that, there are radiation hazards also are there, which give us neural tube defects with cleft palates. So the risk of birth defects, if we see, are multifactorial, environmental and genetic. And in that, we see that folic acid deficiency is one of the leading causes, diabetes, obesity, hypothermia, and many of the mothers are not aware that they are pregnant, because it happens during the first 2 to 3 weeks when the new relation sets in. So the drug abuse and alcohol consumption, drugs, infection, radiation, they are the factors. Along with this, the genetic predisposition is there. In mouse, almost 200 genes have been identified, but in human, it's only 80 genes, and which gene is acting where is also still under research. But environmental gene interaction is the maximum, which is multifactorial, and 55% of the defects. So majority of neural tube defects can be treated by folic acid supplementation before pregnancy, when they are expecting to get pregnant, and throughout pregnancies. So there are a number of challenges that, because of these precise molecular techniques, diagnosis has become very easy. So there's been a decline and improvement in medical care, antenatal checkup. So we have had the incidence is coming down. Developed countries, it's much less in comparison to developing countries. So of course, the prevention assumes, in fact, has much greater role, and therefore one has to adopt strategies to give folic acid supplementation. There should be genetic counseling to mothers who are predisposed to these kind of risk factors, and that has to be done by the obstetrician. So prenatal diagnosis, with the scans available nowadays, it becomes easy to diagnose them. Of course, there is chorionic blood sampling, there is amniocentesis, all the precise molecular techniques. So intrauterine treatment can be done by all these methods. And this is my team, Dr. Babita and Dr. Swati Tiwari, who helped me in doing all the fetal autopsies. Thank you very much for the patient hearing.
Or Dr. Surajit Ghatak. Thank you, ma'am. Thank you so much for giving such a crystal clear presentation and explanation of molecular regulation of neural tube defects, how it happens by the down regulation or up regulation of the genes. Now I invite Dr. Surajit Ghatak to kindly chair the session. Sir, you will receive a message to unmute yourself. Just accept it, sir. On your screen, there will be a message to unmute. Yes, sir.
Yes, ma'am. Can you hear me, ma'am?
Yeah, sure, sir. I can hear you. So, sir, I invite you to chair the session.
Ma'am, thank you, Dr. Savita, ma'am. Thank you so much for such a lucid presentation regarding the molecular regulation of the neural tube formation and the expression of various genes in the formation of the brain and the neural tube. There's a question from one of the participants, ma'am. She says if there has been so much of genetic studies, is there any way or is there any scope of preventing such neural tube defects in future?
Actually, there are a number of transcription factors, genes which have been found. So now they are doing targeted gene therapy. I think it's still in the experimental models they are being doing. But for human, I don't think it has still started. It's only with, I would say, that these kind of therapy which are at present done in very highly specialized centers are where you are doing, there are some incidences of intrauterine surgeries. But of course, stem cell therapies have started and there is some kind of blood transfusions also taking place. But targeted gene therapy is still in an exploratory, is in research mode.
Bab, you have also mentioned about nutritional factors like even alcohol for that matter. And what is the role of smoking, ma'am? Because I see a lot of
yes, actually, yes. Smoking, alcohol, any kind of drug abuse, they all do affect our genetic predisposition. If anybody has any genetic predisposition, and even if these environmental factors, they do cause a lot of defect, teratogenic effects during the first 2 weeks of gestation.
And ma'am, role of folic acid, like you said, it has to be started a few months because that's what most of the book says. Yes. So I feel folic acid given at a later date is of no use because after I have seen a lot of doctors prescribing folic acid after first trimester, but I don't think it has got much role after first trimester. And what's your opinion?
Yes, actually, folic acid, which is being used, it's used as a preventive. B12 and folic acid, all pregnancies should be given folic acid and B12 prescribed by the gynecologic obstetricians. But what has been seen that there are certain folic acid resistant cases where demethylation of the folic acid doesn't take place. So when this doesn't take place, so they do not even if they have very high levels of folic acid in their blood, they still do present with neural tube defects. And with this very strong genetic predisposition to neural tube defects, they have been seen that they can be given something they have to be intervened separately by giving them inocetal, which also has a role in prevention of neural tube defects. So this is only with subsequent counseling that one can actually do. These are all very, very elementary. And what we do out here in our department is very elementary, just doing autopsies, finding out the malformations, and just trying to deduce from literature what's wrong. So it's,
Do you follow it up with any genetic study?
we follow it up, yes. In fact, 4 days back we had a fetal autopsy workshop with the obstetricians, so we do have that. They were conducting it and we were showing our finding, and we were seeing that how it matches. We had a body ball defect workshop with them.
And ma'am, last question, ma'am. Suppose we screen this mother, what is the role of alpha fetoprotein in screening?
Alpha fetoprotein, yes, it's supposed
It's a good marker.
Yes, it's a good marker for raised in the blood and in the amniotic fluid, that if there's raised alpha fetoprotein, it gives an indication for anencephaly. But that's not the only neural tube defects that one comes across or encounters. Most of the neural tube defects are now picked up by ultrasound. So ultrasonography with the level 1, level 2 scans are with the Dopplers are very good to pick up all the neural tube defects.
Thank you so much. Thank you for such a lucid presentation again, ma'am. It was indeed very, we learned a lot from this. Thank you so much.
Thank you so much for giving me this opportunity. I did have to rush through because I did waste a lot of time at the beginning. I'm sorry for that, and hope you have a wonderful conference. And I thank the chairpersons and the organizers for giving me this opportunity. Thank you, Dr. Jyoti. You've been amazing.
Thank you, ma'am. Thank you so much. Thank you, Dr. Surajit Ghatak, for chairing the session and accepting our invitation. It was very nice of you, sir.
Thank you so much, ma'am.
Now, on behalf of IVACON 2021, we express our heartfelt appreciation to ma'am and present a certificate of appreciation to ma'am. This is for you, ma'am. We will soon share this certificate. We'll mail the certificate to you. So now we come to the end of the session, morning session, and I hope all the delegates and faculty members and students have enjoyed this session, and I'm sure that they have, that there are valuable words of wisdom for them to carry home. With this note, I finish this session, and thank you, Dr. D.R. Singh, thank you, Dr. Sabita Mishra, thank you, Dr. Ghatak, and thank you, Dr. Sharif, for being with us this morning. And all the delegates for joining us so early on Sunday morning. Thank you all. With this, I request everyone to join the break rooms in which the sessions have started. Dr. D.R. Singh को unmute करें. Sir, you have been unmuted. You wanted to say something, Dr. D.R. Singh?
No, I just wanted to congratulate Dr. Savita Mishra.
Okay, sir. Thank you, sir. Thank you, sir.
Okay.
So we'll close this session and we'll move for the oral presentations. Thank you all.
dear delegates who are right now in the main room, they are requested kindly join the different room where the scientific session is going on. One is from 10:30 to 11:45 a.m. and other one is from 11:45 to 12:45 a.m. So all of you requested kindly go through, break down the room and enter into the different rooms and enjoy the scientific session. Thank you very much. Hello everyone. Good morning. It is my humble request. Kindly go through the breakdown room where the scientific session is going on.
I request our guest here to kindly join the breakout room for the sessions, as the oral presentation is going to begin very soon. Thank you. Thank you so much. I request our audience here to kindly move to the breakout rooms, and if they are finding any problem related, to move towards the breakout room. Let us know. We are here to try to solve out your problem. Thank you.
जी जिसको कॉल करो बोलो कि रूम नंबर
चलता रहेगा यार।
Hi,
Tracy.
Jyoti. How are you this morning? Sorry, shall I put my video on?
How are you?
Hi.
It's early morning for you. Hi.
It's a very early morning for me, but it's fine. It's no worries. Hi, Cecilia. How are you?
Hi, Cecilia. So, actually, there is nothing going on in main hall right now. It will begin at 12:45. Right now, the breakout sessions, they are working. So you can go to the breakout rooms. There, the presentations are going on. So we'll be starting here, session right at 12:45.
So that's in 12 minutes time, roughly.
Yeah, around 15 minutes.
Okay, great.
Within
No, that's
15
perfect.
minutes, we'll start this session.
Sorry?
Within 15 minutes, we will start the session in main hall.
Excellent. No, that's brilliant. Thank you so much. Thanks a lot.
Okay, okay. So our chairperson, Professor Rajan Bhatnagar, is also with us. You can see him.
Morning,
Unmute.
Professor Rajan. Good morning.
Hello, good morning.
Sir has been unmuted.
Sir, send a request to unmute.
Yeah.
Good morning, Tracy. How are you?
I'm very well, thanks, Rajan. How are you?
I'm waiting to hear from you.
I'm delighted to be here. It's really nice, even though it is a little early in the morning over here.
I know.
It's good for the soul to get up at really early on a Sunday
Get up in the
morning.
morning, right. So I'm closing this hall right now,
Yeah.
and we'll meet again here at 12:45. Okay?
Okay.
Fine.
Thank
If
you. Bye-bye.
If you want to move
Bye-bye.
around the breakout rooms, you can move around and see the sessions which are going on there.
Okay, great. Thank you so much, Jyoti. Thank you.
Cecilia, same for you also. You can also move around the sessions which are going on
Yeah.
in breakout rooms. Okay?
Yeah.
Okay, okay, sir.
Hello.
Hi, you wanted to say something?
Oh, no.
Again, I was catching your sign language.
No, I was just going to say hello to Tracy, but I've managed to chat to her in the chat, so that's all right. I just came to support her. It'd be nice to see her, to hear her lecture as well.
Yeah.
Thank you for sending the link. Yeah, because I wasn't sure whether we would have the link for today as well. So when you sent it, I thought, oh, well, I must come.
Tracy is live now.
Yes, sorry, they muted me, Cecilia. I mean, I can understand why, Jyoti. Sorry, I'm not complaining, but I couldn't say hello, Cecilia, because I was muted and I was unable to unmute myself.
Yes, yeah. I mean, you've organized it really well. I mean, it must be a lot of work for you.
Yeah, it was. It was.
I can
work.
imagine. Yes. I mean, I see you have a lot of teams. You have IVA team 1, 2, 3.
we have different teams for different sessions, for different oral presentations. So we have a handful of residents with us, the PhD students. So they have made their teams, and the faculty members are also involved with them. So we have different teams, and we have given them co-hostship. So they are managing the presentations in breakout rooms. So right now we are having oral presentations, and in the afternoon we'll be having a set of poster presentations.
I see.
Yeah.
Yes.
Okay.
Yeah, I see that yesterday there were about over 100 participants. Is that right? Or?
Yeah. Some participants were here, some participants were in breakout rooms. Because of this technical problems, our session got delayed, and the persons who were in breakout rooms, they were attending the session, they were presenting themselves over there. So in the main hall, we had not that much of a strength because of the simultaneously going on presentations.
I see. Yes. Great.
Okay, we'll see you in about 5 or 10 minutes.
Sure. Have your tea.
I'll hide as well then.
Thanks, Jyoti. See you shortly.
Okay. So, welcome again to the scientific feast. We are back here. Now, on the behalf of Team IVACON, I welcome Dr. Tracy Wilkinson, guest speaker for this session, distinguished chairpersons, Dr. Renu Dhingra and Dr. Rajan Bhatnagar, respected teachers, and dear delegates. With the advent of newer surgical methods and interventional radiological techniques, the improved methods of surgical training are needed. And among the various methods which are available, the simulating surgery on human cadaver is closest to the actual practice. But for this, we need soft cadavers because they give lifelike feeling to the surgeons. So our today's guest speaker is going to enlighten us about solving clinical problems with anatomical solution. So welcome, Dr. Tracy.
Morning, everybody.
Just a minute.
Would you like me to share my screen?
No, not right now. I'm just going to introduce you.
Sorry.
For our participants. So Tracy is Principal Anatomist, Cox Chair of Anatomy, and Director of Center of Anatomy and Human Identification at University of Dundee. She was former Associate Dean for Recruitment and Admissions at School of Science and Engineering in biomechanics and anatomy, and worked in several countries in different capacities, like Germany, parts of Africa, and UK. And in UK, she worked in England, Wales, Northern Ireland, and Scotland. She is really interested in learning and teaching and has got many years of experience of teaching undergraduate, postgraduate, medical, dental, and paramedical students. She has publications in various fields like medical, anatomical education, biomechanics, clinical anatomy, functional anatomy, and human variations. She runs International Anatomy Training Program for Young Scientists, which is hosted by Anatomical Society and American Association of Anatomy. She was there in organizing committee of conference that was held under the flagship of IFA in London in 2019. So I welcome Tracy to this August gathering, and the chairpersons for this session, they are Dr. Renu Dhingra and Dr. Rajan Bhatnagar. Dr. Renu Dhingra is professor at AIIMS New Delhi, and she is basically doing her research in the areas of plastination, molecular aspects of placentation, biomarkers for preeclampsia. She is interested in standardizing the soft embalming techniques in India and in designing new indigenous polymers. She has received several awards like Professor Hirendra Kumar Chatterjee Memorial Gold Medal, Dr. G.P. Talwar Gold Medal, and Dr. Dharam Narayan Gold Medal. We welcome you, Dr. Dhingra, to this session. Our another eminent guest chairperson for this session is Dr. Rajan Bhatnagar. Dr. Rajan Bhatnagar is heading the Department of Anatomy at Ram Manohar Lohia Institute of Medical Sciences at Lucknow. He is on National Editorial Advisory Board of Medical Journal of Armed Forces India. He has successfully established anatomy department in 3 medical colleges, and he has been nominated for bringing adaptation to students' edition of Gray's Anatomy 2012. And he is also nominated as a member of advisory board for reviewing 5th edition of Knitter's Atlas of Human Anatomy. He's resource person for a number of courses and workshops that are conducted by the Department of Medical Education. We welcome you, sir. Now I would request Dr. Tracy to deliver her talk on her topic on Thiel-Zimbabwe. Welcome you, Tracy. I would request you to share your screen.
Oh, thanks. I've got host disabled participant screen sharing. So can you allow me to screen share?
Yeah, sure, we'll do that.
Thanks. Ah, brilliant. Thank you.
Okay.
So can you see my screen? I'm just going to full screen.
Yes, I can see your screen.
And is that on full screen, Jyoti?
Yeah, it is on slideshow now.
Excellent. Thank you very much indeed. Okay, so good morning, everyone. I'm really pleased to be here, and thank you for a very nice welcome and introduction from the IVACON team. I'm absolutely delighted to be here. My topic, as Jyoti pointed out, is solving clinical problems with anatomical solutions. My name is Professor Tracy Wilkinson. I'm from the Centre for Anatomy and Human Identification at the University of Dundee, as you've just heard. So first of all, I'd like to say a big thank you for the invitation. I'm delighted to be here. Of course, it would have been lovely to have been in Lucknow in person, but in these COVID days, it's impossible to travel anyway. But I'm really, really pleased to be at this lovely conference, and by all accounts, it's been going really, really well. So thank you so much for the invitation. I just wanted to show you where Dundee is, because you may not be aware. People have heard of Crocodile Dundee, but we have nothing to do with Australia, I assure you. That's Dundee. We're up in Scotland in the UK, and I can assure you it's much earlier in the morning and possibly quite a lot chillier than it might be where you are at the moment while we're here. But there you are, Dundee, where the big blue dot is. I'll just show you Scotland, a little taste of Scotland. It's a very beautiful country, and if any of you ever want to come and visit, please do get in touch. I'd be delighted to welcome you to our very beautiful country here in Scotland. So here's a little taste of the country in which I live. So that was just a little taste of Scotland to see how very different it might be from where you're living at the moment. Please note, I am going to show you quite a few cadaveric images, and I just wanted to remind you that in keeping with UK legislation and the ethical procedures here, our donors have given their bodies for medical education and research only, but not for public display. So I just ask, or remind you, should I say, these pictures are for the purposes of this conference only and not for general distribution, and certainly not for public display or social media or something. So I just wanted to put